Good evening. Tonight we will talk about transcription in prokaryotes. I thought the McGraw-Hill textbook on genetics had some great diagrams displaying this process, so we will talk through those. The overall process of transcription is 3 stages: initiation, elongation, and termination. This results in the formation of an RNA copy from the template strand of DNA and can be seen in the first half of this image. We will ignore the polypeptide formation for the intents and purposes of this lesson, which is the entire row of translation. To emphasize from the beginning, the RNA strand is built in a 5' to 3' direction, but read in a 3' to 5' direction. This means that new nucleotides are added to the 3' end. In initiation, an RNA polymerase binds to the promoter or starter region of DNA and begins to separate the double strands. The RNA polymerase is composed of protein subunits, and notably when it interacts with what's called a sigma factor, can be called RNA polymerase holoenzyme. After separating the strands, we can more easily see a template that can be read in elongation. In the second step, the RNA polymerase moves down the template and reads each base. This helps build complementary RNA nucleotides into a chain, as seen in yellow. If we were to go back to this first slide, you may be able to note that the bases of the new yellow transcript are the same as the red coding strand, aside from replacing thymine with uracil, but is also complementary to the blue template strand. Just to reiterate and/or summarize the importance of the start of transcription and its direction, the bottom image shows initiation in bacteria. The promoter is located on the 5' end and is composed of a 35 region and 10 box, also commonly known as the PRIB-NOW box. These regions are significant as they have the quote-unquote instructions for the RNA polymerase. The 35 region is recognized by the polymerase and helps determine the binding of the subunits, and the 10 box helps the base separation process of the DNA template. In a theoretical scenario without the 10 region, the rate of transcription could be affected because no new bases could access the template strand of DNA. An important function of the 10 box is the composition of thymine and adenosine because they're easier to break apart. This diagram, albeit came from Wikipedia, is really interesting because it shows how proteins that would normally activate or inactivate the promoter region demonstrate different effects when they are present or absent. Not only that, but the location of inactivation is also in play. Consider that B is the 10 box, so without it, as seen in the middle to bottom diagram, there would be a very low level of transcription. And last but not least, termination would occur. So another sequence of DNA called terminators signal that the RNA transcript length is now complete. Intrinsic terminators trigger what is seen as a hairpin loop located at the bottom left, and then the RNA polymerase is released. The single-strand mRNA can now move on to translation, and the double-stranded DNA can be reused for further transcription.